Hello dear viewer, welcome to my YouTube channel. In this video lecture, the topic under discussion is Gramstaining procedure. This whole procedure will discuss in this lecture that what is Gramstaining and how we stain bacteria through different dyes or through which procedure bacteria will stain. Let's start the lecture. Before starting the Gramstain procedure, we must know the difference between gram-positive and gram-negative bacterial cell wall composition, because gram-staining is totally dependent on gram-positive and gram-negative bacteria cell wall composition. Now have a look at gram-positive bacterial cell wall. This mesh-like structure is peptidoglycan layer, which is thick in gram-positive bacteria while thin in gram-negative bacteria. Because of this reason, gram-positive bacteria will retain the primary dye, that is crystal violet, while gram-negative bacteria have thin peptidoglycan layer, as well as they contain an outer membrane layer. As the outer membrane layer of gram-negative bacteria dissolve, that's why gram-negative bacteria does not retain the primary dye and they washed out. The reason behind this is that gram-positive bacteria retain primary dye when they combine with iodine. They make crystal violet and iodine complex, while gram-negative bacteria will lose primary dye upon decolorization. Well, before starting work at lab, you must wear personal protective equipment, gloves, lab coat, mask, etc., which aim is to protect himself from hazardous or toxic or pathogenic microbes. Bacterial smear formation or bacterial smear preparation is the first step in Gramstaining, in which we use clean and grease-free slide by using either liquid media or solid media. Liquid media in the form of broth or solid media in the form of nutrient agar or McConkey agar. First, we pick bacterial colonies from liquid or solid media and spread it on the slide surface. Let them air dry and then heat fix through flame. The purpose of heat fixation is that the slide must pass further from few steps using different dye to prevent the smear loss. In the second step, the slide will flood using different dyes that passes from few steps, and at last observation through microscope, which aim is to confirm the bacteria is either gram-positive or gram-negative on the basis of color. For Gramstaining procedure, we have four types of dyes known as Gramstain reagents. These reagents include crystal violet solution, Grams iodine solution, ethyl alcohol 95%, and Safranin dye. First step of the procedure is crystal violet staining. In this step, heat fixed dried slide is taken and pour the slide with crystal violet dye for 1 minute to stain the whole smear, which is covered with crystal violet. After the slide has been trapped with crystal violet, it is washed off under tap water slowly. Second step is staining with Grams iodine solution. In this step, we add Grams iodine solution on slide, which act as a mordant. By mordant, we mean that it gives strength to the primary dye and anchored them inside the bacterial cell wall. It forms insoluble crystal violet iodine complex, which imparts purple color to the cell. In this step, we flood the slide for 1 minute and then wash with tap water. Next is decolorization step. In this step, we use ethyl alcohol 95% for 5 seconds. We add ethyl alcohol drop by drop for 5 seconds. By doing this, gram-negative bacteria completely lose primary stain, but if bacteria is gram positive, they will retain the primary dye. The function of 95% ethyl alcohol is that they dehydrate the bacterial cell, which leads to shrinkage of the thick peptidoglycan layers and hold the stain strongly. In fourth step, we add safranin or counter stain. By counter stain, we mean that this stain differentiates between gram positive and gram negative bacteria. If the bacteria is gram positive, they does not retain safranin because they already retain the primary dye, while gram negative bacteria lost their primary dye upon decolorization, so that's why they retain the safranin dye. So by this way, color differences appear between gram positive and gram negative bacteria. In this step, we flood the slide with safranin for 1 minute and then wash with tap water. After the procedure is complete and the slide is ready for examination, then we use microscope, that is oil immersion lens, for examination. Oil immersion lens have 100x magnification power. We put a drop of oil on slide and then observe. The oil immersion lens have same refractive index on slide, so the microscopic light does not deflect and enter directly to the ocular lens or objective lens. That's why image resolution is more better than other lens. During microscopy, the image will appear in microscopic field after gram staining either pink or purple. Gram negative bacteria will appear pink and gram positive bacteria will appear purple. So this differentiation is based on color. That is the main difference between gram positive and gram negative bacteria. Thanks for watching. Don't forget to subscribe, like, share and comment.